Howdy, folks. Thanks for checking into Mr. Ulrich's Land of Biology. I, of course, am Mr. Ulrich. In this video, we're going to continue our discussion on sexual reproduction and get into the form of cell division that makes sexual reproduction possible, and that's meiosis. Now, to recap the important points that we've already kind of discussed about sexual reproduction, the big hallmark of sexual reproduction is not the number of individuals that are required in order for it to take place. It's not that it's two parents in sex and one parent in asex, because now we know that there are plenty of instances of organisms that can make both gametes, and those gametes can fuse and make another organism sexually, even though there's one parent. So the big hallmark of sexual reproduction then is the fusion of nuclei, fusion of cells. And that means that we're going to have a special form of cell division that's going to yield cells that only have one half of the genetic component from each parent, so that you can put those together and fuse them in order to form your first somatic cell, your first body cell, called a zygote. Now, of course, like I said, it's not going to be mitosis that makes this, because if we have mitosis make these cells, then you're going to be adding full sets of genetic material together, and then your first body cell will have twice the number of genes that it's supposed to have, and that is a problem. This means then that we can categorize our cells into kind of two broad categories. We have our body cells, those somatic cells that you find kind of all over your body, and they all have the same set of genes. Right? We have all of our body cells, all of our somatic cells have all the same genetic material. The difference from one specialized cell to another specialized cell is which genes are turned on and which ones are turned off. Now, our gametes are different from this, right? These are the sex cells. These are the ones that are going to fuse, and they contain half of the chromosomes of a somatic cell. Now, once we start counting chromosomes, we bring up this term we kind of roughly referred to as ploidy, and we start to refer to the number of chromosomes. We'll talk about sets here as we get going on. We get into homologous pairs and stuff like that. But this is where, like I said, where we're categorizing or typifying the number of chromosomes that you find and how they're arranged in that organism. Just to be clear on a couple things. First off, I'm sensitive to issues of sexual identity, gender identity. I recognize those, that there are variations on the theme that we present as most general and traditional. So when I'm going to fly in here, when I'm talking about sex and sex determination, I'm talking, like I said, most generally, traditionally as determined at birth. So just to clear up some vocab things here. I'm trying to get my animation to come up. Here we go. Gamete, we're referring to those sex cells. The male gamete is the sperm. It is motile and generated. They're generated in the testes. The term gonad we can use. That's not a bad word. It is a technical term, and it's also not reserved for males as well. The female gonads are our ovaries, and this is where the ova are produced. That's plural of ovum. And my favorite joke, of course, if you go to the diner now, you can ask for your ova easy. Hardy har 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 har. There's an ovary for you. The goal of those gametes is to fuse the sperm and egg to form your first somatic cell called a zygote. So our egg with 23 chromosomes fuses with the sperm with 23 chromosomes to create that zygote. It's 2N because now it has a pair of each chromosome. It's going to be important here in a second. So all of your body cells, your somatic cells, all originate from that first zygote. As I said, we're going to talk about ploidy. and how we, like most sexually reproducing organisms, are diploid, meaning that our chromosomes come in pairs, so sets of two. And as Mendel noticed, you get one from one parent and the other from the other parent. Like I said, this is the way that it happens in most organisms. And that gives each one of us, each species of organism, a typical chromosome number. Now, that doesn't mean that there's one chromosome number for each organism. We'd be pretty limited. There's lots of doubles, right? Us and mice both have 46 chromosomes. Our closest relative, the bonobo, has 48 chromosomes. So just arranged differently, even though we're a lot closer related to them. Sometimes it is helpful for us to be able to diagnose if a developing fetus has a abnormality in its number of chromosomes. That can happen through meiosis. I'll show you where that happens in a bit. But the way that way that we can test for this is through a process called amniocentesis. This is where we take a needle and go right through the abdomen of mom and get into that amniotic sac and withdraw some amniotic fluid and get some cells from the baby. Spin those down and culture them so that they'll start to divide. Take a picture of a cell that's dividing. That way we can have those chromosomes. And from that picture, we can develop a karyotype. And this is the karyo again. That's the old Greek prefix for nut or kernel. So we're talking about the nucleus. We're talking about the genetic material within the nucleus. And we can see, count them up, right? Match them up by size and banding pattern, and count them up and make sure that there is the proper number in that cell. So this brings us to this concept of homology, homologous chromosomes, that we get one from each parent, right, and each homologous pair. Hopefully you've been talking about this in your genetics class, in regular bio. In the genetics unit. And so, like I said, you get one, the maternal one and the paternal one for each one, each set. So this is where we get this concept of homozygous and heterozygous from, right? How you can have two of the same gene for eye color, right? Both brown or both blue, or they can be different. Well, the reason why that can happen is because you have two chromosomes that each have the same traits encoded on them, eye color, even though sometimes those traits, those genes, can be slightly different from one another in the case of brown versus blue. We talk about that position on a chromosome as a locus, where we're going to find a particular gene. So at each locus on those two chromosomes, you're going to find the same characteristic, although it might be a different allele. That means then that we have 23 pairs of homologous chromosomes. Well, if you are a girl, traditionally as determined at birth, then you have 23. If you're a boy, you have 22 pairs of homologous chromosomes and one pair of non-homologous chromosomes. What am I talking about? Well, we all have 22 pairs of autosomes. Those are our body chromosomes, as we call them. And then there's these two that get referred to as the sex chromosomes, because the way that, like I said, it typically works. It's whether you have a Y chromosome or not that's going to determine whether you develop towards maleness or not. During meiosis, these homologous pairs of chromosomes are going to join up, and they're going to form a single unit called a tetrad. And the reason why we call them a tetrad is because they have 1, 2, 3, 4 different chromatids. Remember, these sister chromatids, right? Here's dad's two sister chromatids, are the result of replication. And so if dad has blue eyes, then on this chromosome at this locus might be a blue eye-ness. Since the other one here is the result of replication, it is also going to have blue eye-ness. Mom, on the other hand, she might have brown eyes. But these two alleles here are both going to have the same chromosome, the same allele, because they are the result, again, of replication. Let's talk a little bit about how these sex chromosomes work, because they're peculiar in the way that they function. We're tempted to think that because we call them sex chromosomes, that these would be where the genes for testosterone or estrogen or PPs or hoo-has or whatever are going to be located, and that's not exactly how it works, really at all. Now, most traditionally, again, most often and most generally, if an individual has two of the same chromosome at the last chromosome there, the 23rd pair of chromosomes, what we call the sex chromosomes, that individual will present as a female. Um, if it's XY, which is non-homologous, and I'll explain what that means in a second, then the individual typically presents as a male, typically. There are, again, variations on this theme. Now, the way it works is there is a small portion of the Y chromosome called the SRY, or sex-determining region of the chromosome, of the Y chromosome. And if an individual has that, then they tend to present as male. Now, we know that crossing over can take place, and that there can be some jumbling that goes on. We know that pieces of chromosome can break off, so it's completely possible for a part of this Y chromosome that has the SRY to break off and attach to a different chromosome, not necessarily the X chromosome, but maybe chromosome 9 or whatever. And in that case, the individual won't have necessarily a Y chromosome, but could have a present as a male because they have that SRY somewhere else. Now, that's not the only way that we end up with intersex, but it is one way that we can have an individual. And I'm not sure that that would even be considered intersex, but that's the one way that we can have individuals presenting differently from what their chromosomes might indicate. So there's those sex chromosomes down there, and like I said, again, most traditionally, this individual with two of the same sex chromosomes would present as a female. Now, some other terms that we should be able to use, that means our cells, since they have our body cells, since they have genes that come in pairs and chromosomes that come in pairs, that we are diploid, right, di for two. And then when we create these gametes, those gametes have half the number that they normally would have. And so that's why we call them haploid. We can also call them monoploid is another term. Now, the Regents used to make a big deal out of this. I haven't seen it in a while, but they like to ask about if gametes were made mitotically, what would happen? And of course, like I said, there'd be a doubling of chromosomes, and you got to have the right number of chromosomes. It doesn't really work very well when you have extra chromosomes or are missing chromosomes in most cases. Now, this is why we call meiosis reduction division, because we can achieve this halving of the chromosome number. The cool thing about meiosis, on the heels of learning mitosis, is that we're going to use the exact same machinery that the cell uses when it goes through mitosis. In fact, it uses the same steps, and they're going to look exactly the same. And the way we're going to get this halving and be able to make it work is to actually stack two kind of rounds of mitosis on top of one another. We call it meiosis 1 and meiosis 2, and there's a couple little differences that make it very important in differences. And there's no replication of DNA in between the two. It's going to happen, like I said, in the same prophase, metaphase, anaphase, and telophase. During meiosis 1 is when we actually achieve the halving of the chromosome number. The way this happens during prophase, just like in mitosis, it's going to take a long time because we have to condense all of that genetic material, arrange it into chromosomes, build all those spindle fibers, move the centrioles, connect the spindle fibers to the centromeres. We also have an added step. There we go, chromosomes condensing. My animations are taking a little bit to come here with me. The added step is the formation of those tetrads, right? Making the homologous pairs come together into a single structure. This formation of the tetrad is a process known as synapsis. This is also when we're going to see crossing over take place. Little pieces of one chromatid swap places with the other chromatid. All right, and this is how we shuffle up genes. Yet another way that sex produces such a wide variety of allelic combinations. This means that during prophase, those homologous pairs of chromosomes attach to the spindle fibers. During metaphase, then, when we see these line up, instead of during mitosis, where we would see 46 double chromosomes lining up, now we're going to see 23 tetrads, pairs of homologs lining up individually across the equatorial plate. And there's going to be a variety of different arrangements, right, of maternal, paternal, maternal, maternal, paternal, paternal as they line up of those chromatids, so that when they do segregate, which is going to happen. So now, instead of seeing 46 single chromosomes migrate to either pole, now we're going to see 23 double chromosomes, because now we had 23 things lining up, right? They're going to segregate into double chromosomes, right? And we're going to have 23 going to each pole of the cell. Right, they're still double chromosomes, right? So those chromatids are still attached. Now, they're supposed to divide equally, but sometimes there can be mistakes, right? And this is where we can start to get some chromosomal abnormalities. We can get some chromosomal mutations taking place, right? It's a process called non-disjunction, when those homologous pairs are supposed to separate and they don't do it equally. So you'll get 3 going to one side and you'll get 1 going to the other side. What this is going to do is it's going to kind of jumble up the numbers, and you're going to end up with a gamete that has, instead of 23 chromosomes, it's going to have 24. And you're going to have another gamete that's going to have, instead of 23, it's going to have 22 chromosomes. If those gametes go ahead and are the ones that fuse, and fertilize and become the baby, you're going to have a baby that has the wrong number of chromosomes. And this is one of the reasons why we do, like I said, a karyotype. Now, this individual, if you look closely at the 21st position, they have 3 21st chromosomes. It's called trisomy 21. It's also referred to as Down syndrome. It is one of the few survivable versions of trisomy. Another version is having extra sex chromosomes, both having an extra X chromosome or extra Y chromosomes, and those are definitely survivable. They can alter the ability for reproduction, because it makes it impossible to make homologous pairs. During telophase, then, we're going to see a pinching off, and now we're going to have what are called secondary sex cells. And the problem is, like I said, those cells, though we have halved the number of chromosomes, the problem is they're double chromosomes. And we can't send cells that have double chromosomes into interphase, because they would want to replicate again, and they can't just skip replication, right? They don't think like that, I guess, is the best way to describe it. And so we're going to need to have another round of prophase, metaphase, anaphase, telophase to take these double chromosomes and turn them into single chromosomes in another round of basically mitosis. Since those chromosomes are already doubled, when we go into the second round of meiosis, called meiosis 2, there isn't any replication of DNA. We already have those double chromosomes in position. So it's going to look just like mitosis, exactly like mitosis, except there's half the number of chromosomes. This means that prophase is going to involve attaching those double chromosomes to spindle fibers. This means that during metaphase, they're going to line up across the metaphase plate. This means that during anaphase, those double chromosomes are going to segregate into individual chromatids, and those chromatids are going to start to migrate to individual poles of the cells. Telophase is going to look just like in mitosis, right? We're going to have new nuclei form around those now individual single chromosomes, right? We're going to see the division of the cytoplasm, and we're going to end up with now 4 cells that are haploid with single chromosomes each, right? 1 of each homologous pair of chromosomes, perfect for gametes so that they are set up and ready to fuse. It's important to recognize that, yes, those gametes are genetically distinct. Here's one color combination, right, of maternal, paternal, I'll say. I don't remember which one was which, right? Paternal, paternal, maternal, maternal, right? Paternal, maternal, and depending which ones had the dominant and which ones had the recessive and how much crossing over took place and where the crossing over took place, each one of these gametes is going to be genetically distinct from the other one. This brings us to a couple more terms. We're almost done. Just hang tight. All right, since we're talking about making gametes, the term that we would use for this is gametogenesis, because we are generating gametes. In the females, this is referred to as oogenesis or oogenesis. Yes, you pronounce each one of those O's individually from one another. All right, making ova. In males, there we go, there's some pictures, we're going to produce sperm, so it's called spermatogenesis. But I guess my numbers are off there a little bit. In males, again, we're going to end up with 4 of the same sized cells. They're all motile. They all have 23 chromosomes, right? Spermatogenesis starts right around puberty and continues for the entire life of the male, generally speaking. It's kind of quantity over quality, right? We make lots and lots, males make lots and lots and lots of gametes, but they can be a little messed up sometimes, right? We not have the right number of tails and heads and all kinds of fun stuff going on. Now, oogenesis is going to look quite a bit different. All right, hopefully you can see right off the bat, big difference. All right, one cell is quite a bit larger than the other cell. This little polar body, as it's called, is really just half of a nucleus. Right, all of the cytoplasmic material is going to end up in one of the secondary oocytes, as they're called. Not a term you need to know. All right. And so it's unequal cytokinesis. All right, remember that cell's got to provide all of the cytoplasm for the first somatic cell, so that somatic cell can divide, and it can divide again, stuff that we'll talk about later on. Now that secondary oocyte is going to divide unequally again, and all of the cytoplasm basically is going to end up in one ovum. And half of the genetic material, right, 1 of each of the double chromosomes, 1 chromatid from each one of those double chromosomes, is just going to get kicked out into one of these little polar bodies, right? And so you really only get 1 egg from each primary sex cell, where in males you get 4. Now, I'm going to leave it there. That's enough for now. Thanks for sticking with me as long as you did. If you have any questions, feel free to email me. I'll try to get to them as quickly as possible. Other than that, we'll see you in class.